Hello, my name is Cozy, and my report was on heavy metal content in commonly consumed seaweeds in the US. So, currently, seaweed is increasing in demand. The seaweed industry is expected to grow 8 to 10% annually due to population growth in high consumption countries, growth of plant-based diets, product innovations, and new market penetration. Currently, over 98% of edible seaweed is imported into the US. This is about 16 million dry pounds, as shown to the chart in the chart to the left. The leading importers into the US are Iceland, China, Canada, South Korea, and the Philippines. Only 95,000 dry pounds come from domestic sources. So, seaweed is a source of dietary fibers, amino acids, phenolic compounds, vitamins and minerals, carbohydrates, lipids, and it is the most significant source of iodine. Seaweed is one of the major absorbers of carbon dioxide, water, nutrients like nitrogen and phosphorus, and heavy metals. The environmental conditions of where seaweed is grown, such as contaminants, turbidity, salinity, temperature, nutrient content, light exposure, pH, wave exposure, along with the seaweed species, all influence absorption abilities. Oil, plastic, and industrial and agricultural waste, such as fertilizers, are the leading ocean pollutants. When these substances enter the ocean through methods such as sewage, industry discharge, and runoff, the metals in seaweed may increase from these. This study focuses on the contaminants found in common US edible seaweed brands, and the goal of the study is to determine if consumers should be more cautious about the seaweed they purchase to prevent future health risks, such as varying cancers and other forms of damage to the body. There are 3 classifications of seaweed based on the dominant pigment they use to photosynthesize energy. Phaeophyta is the brown seaweed, Rhodophyta is the red, and Chlorophyta is the green. The species of seaweed that this study focuses on include wakame and kombu, which are both brown, nori and ogonori, which are both red, and finally umibudo, which is green. After dividing these seaweeds into groups and analyzing the ability of each from Amazon and Costco, a random stratified sampling method was used to proportionally test each of the species to avoid bias. A total of 10 wakame, 11 kombu, 16 nori, 1 ogonori, and 6 umibudo were tested, a total of 44 different seaweed brands. Each seaweed was tested 3 different times for a total of 132 quantitative samples. This is kombu or kelp and wakame. These seaweeds have 2 things in common: the carotenoid that turns them brown, and both fucoidan and alginate, a polysaccharide found in the cell walls of only brown seaweeds. Arsenic is attracted to sulfur, and fucoidan contains sulfate groups, which may suggest a potential relationship between fucoidan content and the higher levels of arsenic. When looking at the type of environment these seaweeds are found in, wakame grows in 1 to 3 meters deep and exhibits high tolerance to sunlight and wave exposure. It favors cold water with a preference for temperature less than 53 degrees Fahrenheit and dies in temperatures over 68 degrees. Kombu or kelp inhabits water around 5 to 8 meters deep in areas below 68 degrees Fahrenheit and generally takes around 2 years to reach the level of maturity required for harvesting. Harvesting usually only takes place during the summer months. Nori is mass-produced in calm water such as bays or inland seas and grows around 1 to 8 meters. They are typically stacked on nets, which is why this species may be the perfect victim of pollution. When looking at the harvesting process of brown and red seaweeds, they are first cultivated, harvested, cleaned, minced, then roasted. The nori are typically either processed into sushi-grade sheets or further cut into snack packs. Umibudo came hydrated and in a sealed metal package for preservation purposes. It is grown in shallow coastal water along the Indo-Pacific at milder tropical temperatures, and they are cut and undergo minimal processing. The metals looked at will include total arsenic, cadmium, chromium, and lead. Each of these are referred to as heavy metals because they have a relatively high density and atomic mass, typically above 5 grams per centimeter cubed. We chose these metals because they are most commonly found to induce human poisonings. According to the CDC, these are the first 5 metals to appear on the first 4 metals to appear on the EPA priority list. Connecticut's Department of Agriculture suggests these limits of seaweed farmed in state taken from the French Agency for Food, Environmental and Occupational Health and Safety, or ANSYS. Currently, there aren't any regulations in place by the FDA for seaweed heavy metals. They monitor and test for metals in foods, but that will only result in a Prop 65 label for businesses that sell in California. Notice how arsenic and lead limits are high. These values will be used as a reference during this study. The first hypothesis was that arsenic accumulation would relate more to the seaweed color category than to the environmental levels, and brown would have the highest levels of arsenic contamination. This may be due to the presence of fucoidin or alginate, which is found in the cell walls of brown seaweeds. Secondly, since in pre-existing studies, the other metals, specifically cadmium, chromium, and lead, had mixed results, these were more dependent on environmental factors such as contaminants, turbidity, salinity, temperature, nutrient content, light exposure, pH, age of seaweed, and wave exposure. These factors may affect the degree of interaction between metals and the seaweed cell wall. So to obtain the results, we needed to test the samples, and we did this by first weighing out 1 gram of the dry seaweeds, and for the seaweeds that came hydrated, the green ones specifically, 3 grams were weighed out. The exact weight of each was labeled, then added to a pre-cleaned beaker with nitric acid and heated to 60 degrees. Once it was digested, it cooled to room temperature and then was filtered. Then 1 milliliter of this dilution was added to 9 milliliters of nanopure water, filtered through 0.4 micrometer syringe, and went through ICP OES to be tested. And these are the values we used during this study. Before analyzing the results after the samples were obtained, the quantitative values from the qualitative were then differentiated. The quantitative variables analyzed in the study include arsenic, cadmium, chromium, and lead, each measured in parts per million. The qualitative variables included the biological genera or type of seaweed, the color category each fell under, the origin location, then the store that seaweed was purchased from, the brand, and whether or not they used Proposition 65. This box plot shows each of the quantitative variables. There are significantly higher amounts of arsenic, followed by cadmium, chromium, and then finally lead. In comparison to arsenic, cadmium, chromium, and lead are closer in range. Arsenic has a few outliers under 0.05, chromium has a few below 0.1, and lead has one under 0.03. ANOVA for arsenic, cadmium, chromium, and lead showed that there is a significant difference of color between arsenic, cadmium, chromium, and lead of the seaweed because the p-values were lower than the 0.05 significance level. However, we rejected ANOVA because the Shapiro-Wilk test and Bartlett's test were not satisfied with p-values below the 0.05 level of significance. The Kruskal-Wallis-Frank sum test was then used and revealed a significant difference between heavy metal content among different seaweed colors. When looking at arsenic, brown had the highest values, followed closely by red seaweeds. The left displays a box plot of all the parts per million values, whereas the right displays the mean values. When looking at cadmium, red has the highest values, followed by brown seaweeds. When looking at chromium, red has the highest values, followed by brown. When looking at lead, red has the highest values, followed by brown. The summary table also displays what was shown by the previous slides. Brown seaweeds contained the highest mean arsenic, whereas red seaweeds contained the highest cadmium, chromium, and lead. Green seaweeds had extremely low values. When looking at the relative frequency of qualitative variables, it is revealed that most seaweeds originate from China and Maine. The source table proportions reiterate the chart on the previous slide, revealing that most seaweeds originated from China and Maine. In addition, these seaweeds are only brown and red species. The green seaweeds only originate from Vietnam. However, when completing ANOVA, the p-value was below 0.05, which warranted the rejection of the claim that the seaweed color is related to the source. Most seaweeds were purchased from Amazon and were from the family of brown seaweeds. Most red seaweeds are labeled with Proposition 65, as shown to the left, and the most reoccurring brands found was Ocean's Balance. Overall, the quantitative data reveals that there is a significant difference in mean arsenic, cadmium, chromium, and lead across seaweed colors. Brown seaweeds were consistently higher in arsenic, which may be due to the presence of alginate and or fucoidin within their cell walls. Red seaweeds were higher in cadmium, chromium, and lead, which may be attributed to their growing conditions. Each metal was within the safe recommended AN/SES limits, and values from green seaweeds were consistently the lowest, which may be due to their hydrated state. Finally, there was no relationship between the qualitative variables. Our sources of error include not using dehydrated green seaweed and not having large enough sample sizes. This is why more studies are needed in addition to determining the inorganic arsenic by separating it from the total arsenic. Thank you for listening.